go in with a laser microdissection system and collect all these individual cancer cells, separate them from the healthy cells, and then accumulate enough to go for mutation analysis. My name is Kühnel. I'm a research group leader at the Institute of Pathology here at the Uniklinikum in Aachen. My research interests are actually quite broad. Over the last 10 years approximately, we established the platform pathology of the German Center for Lung Research. The main focus of this platform, or one very important focus of this platform, was that we used lung explants as a basis for our research. So the real human tissue carrying the real information of human lung disease. So especially in the beginning, this kind of work was really tedious. I mean, you have to imagine that you are cutting out with a laser tiny areas of cells, maybe like 20, 30 micrometers long. And for example, if you breathe at the wrong time, this tiny piece of tissue is basically flying away. With MMI, they were constantly asking us where our problems actually are, what we could do better, what a better system needs to have in order to be really meaningful in our work. And so this back and forth always worked really nicely with MMI. We could give feedback on what is working, what is not working. And this all culminated basically in this cooperation project where they were providing prototype parts and we were testing them in order to get better laser microdissection system and to speed up the efficiency of our analysis of transcriptomes of these diseases. Very important for a cooperation or to become customer. The customer service is of course very important, especially the reaction time when there's problems occurring was always really important to us. From that point of view, I can say we had contact on every level with MMI, and at every level cooperation worked really nicely, was very responsive, very fast, and we always managed to basically find a solution to the problems that occurred. The other level was the development of new technologies and new applications. That was of course a completely different thing, but also on that level we were able to basically talk about, let's say the future of methods in pathology. And MMI was very interested in making this a reality. And so we ended up testing prototypes of new laser microdissection systems, new motors, which were better pickup of the samples and so on. And also automated structure detection in order to have a system which can identify certain structures by itself or make suggestions of structures which can then be isolated automatically. Especially when we are dealing with, for example, cancer and diagnostic material. Let's say a patient has some kind of cancer where the cancer cells are in limited numbers surrounded by a lot of cells which are just normal. Then the normal signal in standard analysis completely covers the cancer signal. But it's really important to understand where the mutations in the cancer cells lie. And here the only option, or the best option for the patient, is that we go in with a laser microdissection system and collect all these individual cancer cells, separate them from the healthy cells, and then accumulate enough to go for mutation analysis. The investment in the MMI machine was definitely worthwhile, especially at that time, because it was one of very few machines that was on the market. It proved to be very reliable, easy to use, and it worked with us for many, many years, almost flawlessly. Also later turned out to be a good investment because when we did more applications in research, MMI was there to improve with us the functionality of the system, to have the laser microdissection automated, and by that save a lot of time for the researcher spending on the laser microdissection system itself. What surprised me most was that when we initially started with automated structure detection and questioning whether this could be an option in the system, that they were already working on it. So basically they already anticipated where the market was going and where the questions or future questions would arise, and would already work on it with quite a number of people. Once they realized that we are also interested in these kind of questions and being pathologists, they were keen on getting in the discussion with us on what is really needed, how are the characteristics of certain entities, whether we could help to annotate, and so on and so forth. It was so easy to use this that, for example, students that came for a few months and spent some time with us in the lab were able to learn it. So it was very easy to use it in the everyday routine, and it took minimal effort to teach others how to use it and how to be really good at it.